Good afternoon, or good morning, good evening, depending on where you are in this world. Welcome to this CDDF webinar on long-term outcomes of in utero exposure to cytotoxic therapy, with the additional question, is involving pregnant patients an absolute no-go in cancer drug development? My name is Jaap Verwij. I am a member of the board of the CDDF, and it is my pleasure to co-host this webinar. CDDF is a non-profit organization registered in Belgium that provides a neutral platform to stimulate interaction between stakeholders involved in cancer drug development. We bring together academia voices, pharmaceutical industry voices, regulatory authorities, policymakers, and patient advocacy groups with the aim to improve cancer treatment. If you want more information on CDDF, please visit our website. A few technical aspects on this webinar this afternoon. If you have any technical problems, please report these to CDDF via the chat function, which is on the lower button of your lower part of your screen, likely in the middle. If you have specific questions to be asked to us and to the speaker, you can use the Q&A button, which is also on the lower end of your screen. So please remember, if you want to ask a question, that's to be done through the Q&A button. You can already do so during the lectures. There is no need to wait until the end, but you can also introduce questions at the end of the two lectures that we will have. We will present the questions to the speakers during the fireside chat. For better visuality, you can double click on the Zoom screen to enter into a full screen mode, so you'll have the full screen. Today's program, we have actually two speakers, not only one, but two speakers. First of all, Dr. Agnes Saint-Romand, a former head of EMA, head of International Affairs Division, and Frédéric Amand from the University Hospital in Leuven in Belgium. And Agnes and myself will be moderating the discussion after the two lectures. And then it is my pleasure and honor to introduce our first speaker. Dr. Agnes Saint-Romand is a pediatrician who had an early career in hospital practice and then joined pharmaceutical industry and the French Medicines Agency. She joined EMA in the year 2000 and led key areas including pediatrics, orphan drugs, scientific advice, and SME support, and was a key contributor to the EU orphan drug and pediatric regulations. Later, she served as EMA head of portfolio board division and head of international affairs, and retired from EMA in 2021. She currently is active as an expert and board member in global health regulatory ethics boards, including the WHO, Concept Foundation. She is a member of our General Assembly of CDDF and is involved in the medicines patent pool. Agnes, please, the floor is yours.
Thank you, Jaap. Thank you. Hello, everybody. I'm making a presentation which will be an introduction to the main presentation by Professor Amand. I just want to highlight next, please, the fact that what I'm saying is my own view and not represent any of the organizations I work for. Next, please. So the main point, and I could summarize my presentation, but I will not do it. I could summarize my presentation by one sentence: is we need data of medicines used in pregnant women. This is an ethical need, a scientific need, and there is nothing that can justify not having data. We believe that because of the fear that we protect pregnant women by excluding them from research in general and clinical research in particular. But the example we have in history show this is a wrong approach. And I just want to take the latest example, which is the COVID vaccines. The three first vaccines brought in 100,000 participants in trials. There were no pregnant women. And we know that COVID is more serious in pregnancy. At least some of us know it. And therefore, the vaccine were only introduced late for pregnant women when pharmacovigilance showed that there were actually no issues with the mRNA vaccines. Next, please. Of course, you all know that pregnant women can become ill. They can get cancer. It's not very frequent, but it's not rare. They can also want to be pregnant or become pregnant, a mistiming or incidentally. Some diseases are more serious in pregnant women and require treatment. We also talking about lactating and breastfeeding women. Yes, there are alternatives to breast milk, but next, please. In practice, 1 in 2,000 pregnant women is affected by cancer. I found some figures which show that the odds of cancer at delivery is increasing. Frederick has other figures, so he will present them in more details. And we have a huge number of births. Even in the EU only, we have 3.6 million births. So meaning that this is not a small problem, and the number of pregnant women with cancer is not small. We cannot necessarily wait 9 months to address the issue of cancer. It's not necessarily an emergency, but it needs to be addressed. And there is an ethical duty, a general ethical duty to generate evidence, because prescribing without data is not ethical. And also excluding pregnant women is a contradiction to the ethical principle of distributive justice. Next, please. So I will promote very strongly what a similar project to what we did in pediatrics. We had 50 years of wait for pediatrics where we were told that guidelines would produce information. They never did. So guidelines are not the solution to the lack of data. Unfortunately, we need an obligation, and that MIP, the maternal investigation plan, needs to be a product development that is planned, managed, transparent, and shared cautiously, not waiting for pharmacovigilance to give us the information on the number of catastrophes. We need more data, more research. Next, please. We need to generate data with precaution, of course. But pregnant women are not men, not even non-pregnant women. There are physiological differences that justify to have specific studies. We need more non-clinical data, which are also informing us on the risk we are allowed to take or not take. And we also need to look at the structure of the product. We have recently got a new guideline, I will have one slide on it, to show that we actually can do clinical data, obtain clinical data from prospective trial, and we should be doing it. And of course, then we can get post-approval data to identify rare events, need real-world evidence to show that the general population can be is more representative than the population in trials, and follow up all pregnancy during clinical trials and the infants beyond pregnancy. Next, please. So we have few trials. Just for your information, I looked in 2025, looking at PubMed and with the keywords of pregnancy, cancer, and trials. I found only 140 publications and only one, actually it was by Professor Amand, only one referring to a development of a product in a pregnancy. We need precautions, meaning that we need a DSMB. We need active monitoring of women and their safety. We need to identify the priorities, address first the high priorities, and maybe in the second time, the lower priorities. And of course, by sharing information and the result, we will avoid repetition. Next, please. Breastfeeding, yes, there is an alternative to human milk, but at the same time, we know that human milk is better for children, and Frederick will talk about it, so I'm not anticipating this. Next, please. The ICH E21 guideline, very slow process, because the topic was proposed in 21, agreed in 22, the concept paper came in 23, and now we have the guideline. We were expecting comments in September, so I hope that the final guideline will come early 26. The strong point of this guideline is that pregnant and lactating women should be included in drug development, and ICH is a reference for drug development in the world. But the weak points of this guideline at present is that there is too much focus on safety instead of the benefit risk. It only looks at women benefit. It doesn't look at the pregnancy benefit, the embryo or the fetus benefit, and the cancer is not even mentioned. There is vague language on timing, obligation, needs, There is no mention of DSMB. There is a weak approach to PK/PD. So it is actually leaving the decision to sponsors. And unfortunately, we know that sponsors are not very keen on doing it. So we need to have much better wording. So at least the clarity on the need for data is there. We can also hope that 26 will give us a final guideline. Next, please. So my last slide, and we have the choice between fear or science, and it's time to act. We cannot continue excluding pregnant women. This is maleficence, so it's against a ethical principle if we exclude them from clinical trial. And this is even true for cancer. I know it's not simple. I know cancer is actually the area probably the most difficult, because when you don't want cells to multiply, that's exactly the contrary what you want in pregnancy, that cell multiply. So there is an internal contradiction, but at the same time, Frederick will show you that the situation is not as bleak as it could seem, and we cannot just wait for post-marketing data to know what is happening. So, and just a parenthesis in a way, pregnant women are not vulnerable. They don't lose their brain because they become pregnant. They are autonomous, so they can make decisions, including decision on participating in clinical trials. And they do it, not all of them, but some do it because they understand that for their own health and for that of their future children, this is really what counts. And when you explain research to them very well, they do agree to participate. Over. Thank you. So it's my pleasure now to introduce the main speaker, Professor Amand. Professor Amand is a professor at in Leuven in Belgium, Leuven, and at the University of Amsterdam. I want to congratulate him for receiving very recently, a couple of weeks ago, the FWO Excellence Award, a 5-yearly award for clinical research, which is from the National Flanders Research Organization. He's also a honorary professor in South Africa. And you can see his training has been about gynecology and oncology, both in Belgium and in the Netherlands. And we are happy to have him presenting his extensive work on following children having been exposed to anti-cancer drugs. Frederick, your turn.
Hello. Welcome on this webinar. Today's topic is cancer during pregnancy and follow-up of the children. My take-home message for today is that I want to show you that most cancer treatment options are possible during pregnancy, that they need to be treated in multidisciplinary settings, in referral settings, and I want to convince you that more pharmacological studies in pregnant women are in fact highly needed. The incidence on cancer during pregnancy, as you see here of the Swedish registration study, is about 1 in 2,500. In yellow are women as the age group of women older than 35, and you see that this is increasing over the years. And we see it so, or the conclusion is that maternal age contributes to the increasing incidence. So it's good to discuss this because the incidence will increase. I will skip this one, but the cancer during pregnancies are in fact cancers that occur in young women of reproductive age, which is breast cancer is the most common, followed by hematological malignancy, cervical cancer, ovarian cancer, and then all the others. So the main message here is pregnancy per se does not predispose to any particular cancer. No, we see the cancers that occur in young women. But there is a second reason why the incidence will increase, and this is the NIPT test. Here we see deletions and more DNA throughout many chromosomes, and this is the signature of cancer. So this NIPT not only detects actually the trisomy 21, but also screens actually, or can screen the maternal blood for maternal cancer, as in this case, where this patient with this abnormal NIPT, in fact, on MRI had a lymphoma. So we see this more and more in our clinic. We have at least one case per month where an asymptomatic patient does a NIPT, and the NIPT suggests actually a maternal cancer while she has no symptoms. And so that is a second reason why the incidence will increase. To give you a flavor of what we do and what is possible, I opted to share with you some clinical cases. Here we see a patient with, at the left, we see this cervical cancer in a patient with a twin pregnancy. And the previous pregnancy was also a twin preterm with a preterm delivery. So this was really a high risk pregnancy, but the patient was really motivated to continue the pregnancy. So we decided to do an extraperitoneal lymphadenectomy. This here is actually a view of the left pelvis. This is the vein, the artery, the nerve. Also showing that we can easily operate during pregnancy. Lymph nodes were negative, so we continued with chemotherapy. And here we see the response at the right after 3 cycles of paclitaxel carboplatin. So it is chemosensitive as well. We continued in this case with 5 cycles in total, and we then did do a cesarean section at nearly 35 weeks. So this was really a challenging case, and we could take care of the mother and take care of the fetus at the same time. Most common cancer is breast cancer, and we must say that we are frequently, we miss actually an early diagnosis. Patients present with a locally advanced disease. We see here already in inflammation of the left breast. So this is inflammatory breast cancer. You can see the difference here, actually suspect an enlarged lymph node already. Obviously, this is an advanced case already. Even non-experts can see that. This patient actually on clinical examination, it was not so suspect, but then on MRI, you see that a large part of the breast actually is replaced by cancer. So we need to tell doctors and patients that we should do examinations on time, because the pregnancy actually may mimic cancer symptoms, and that can lead to a later diagnosis. And in breast cancer, this is in many cases the case, but not in this clinical case. She was 33 years old, and here the NIPS actually again showed, you see these genomic differences throughout on many chromosomes. And again here, this was suspect of a malignancy. But it was different because while we suspected a breast lesion, on mammogram, actually it was normal. This is a mammogram which is normal despite the fact that there is a cancer. But when the message here is that ultrasound is actually much more sensitive. And we saw here in the breast and in the axilla already lymphadenopathies that showed to be breast cancer. Then we did do this MRI of the whole body, where you see here the pregnancy, but we see also the malignant breast tumor and the multiple axillary and infraclavicular diffusion restricted adenopathy. So also here again, locally advanced breast cancer, which was also HER2 positive. So the patient really wanted to preserve the pregnancy. We decided to give neoadjuvant chemotherapy, 4 cycles of epirubicin cyclophosphamide dose dense, followed by paclitaxel. And although the tumor was HER2 positive, we could not give Herceptin, and I will tell you in a minute why. So that was a deviation from the standard care of treatment. Finally, she delivered of a healthy baby and continued the treatment afterwards. So we could give chemotherapy in both cases. These are the guidelines on chemotherapy during pregnancy. It is important to maintain the dose intensity, to use published standard protocols, to dose according to the actual body weight. There is no reason to increase the dose, and it is recommended to discontinue chemotherapy at approximately 35 to 37 weeks in order to allow the mother and the fetus to recover from the chemotherapy and to allow a safe delivery, both for mother and child. Why can't we give Herceptin during pregnancy? Well, Herceptin blocks the HER2 receptor, and HER2 receptor is also present on the fetal, in the fetal nephron, and that will result in reduced urinary production. That means less amniotic fluid. That means that at the end of the day, there will be no amniotic fluid, and the baby will not be able to ingest the fluid, which the fetus actually needs to develop the lungs. So if you continue with Herceptin, resulting in anamnion, the baby the neonate will die from neonatal asphyxia. Although apart from that, there will be no congenital malformations. But that is the reason why Herceptin HER2 blockade during pregnancy is not possible. We can give chemotherapy. We can also give the supportive drugs with one exception, and that is mainly on the steroids. Because dexamethasone is contraindicated in the first trimester, and also attention deficit disorders are reported with its use, and also with beta-metazone use. That's why methylprednisolone is the preferred option, because it is metabolized in the placenta and is not crossing actually to the fetal compartment. When we give this during pregnancy, we follow up carefully the growth, because we know that the growth can be affected. Luckily, in this case, we see a slight deviation, but thanks to the Dopplers that remain good, we could continue the pregnancy and continue chemotherapy because the growth was not affected too much. We also look to the cervix, because patients with chemotherapy are a bit high risk for preterm delivery. But here we see that the cervix, actually 38, 38, and 33, actually remains well. The length is sufficient, so it's not predictive of preterm labor. So that allows us to continue with the chemotherapy. We planned an induction at 37 weeks, but the patients went into spontaneous delivery at 64 weeks, and actually had an uncomplicated delivery, and also the placenta was normal. This slide shows actually the options that we can have during pregnancy. From the second trimester, we can give chemotherapy. Radiotherapy, it's mainly the first trimester and partially in the second trimester. We can operate both the breast and the sentinel node procedure throughout all trimesters of pregnancy. On the contrary, radiotherapy of the pelvis or even of the upper parts of the abdomen in the second phase of the pregnancy, and anti-HER2 treatment, endocrine treatment, are actually only safe after delivery, cannot be done during the pregnancy itself. So, but the main message is most treatment options are possible during pregnancy. From pharmacological point of view, it is interesting to know that the increase in the plasma volume results actually in a hemodilution. We tested here 4 drugs, and this is the 0 line. We see that for all 4 drugs, the AUC and Cmax actually are lower during pregnancy. Of course, that results in mothers that have less side effects. But what is the oncological consequence here? We don't have so many data, but we have data. And this is in breast cancer. And we see that the group with chemotherapy and without chemotherapy, actually, there was no, the patients compared to non-pregnant patients, but all receiving chemotherapy, that there is actually no difference between the two. So this chemodilution is not affecting the prognosis. Also, for patients with Hodgkin lymphoma, we see that there is no difference. So that the chemodilution is not affecting the prognosis. And of course, this is also important for the maternal consequences. Important is during pregnancy, to consider the thromboprophylaxis, because we have cancer, we have the pregnancy, and we have some surgery sometimes. So those are 3 risk factors for thrombosis. So we should consider the prophylaxis in those patients. Very recent news is that we were all educated that we need, during imaging examinations, we need to protect the fetus with a lead apron. But we have now shown that this is actually making it even worse, because this lead apron makes sure that these scatters, internal scatters, actually they bounce back into the fetus. So that's why we should not use this lead apron anymore, allowing actually or preventing this backwards direction of the radiation. So this is very novel from a few months ago that we published on that. So that's something new that we should inform our patients and colleagues. We're pushing the boundaries. I will not go into detail, but here there's a case report where a mother had 2 cancers, also detected by NIPT, and we were able to treat both cancers during pregnancy. Mother and child are still fine. Very interestingly is the role of immunotherapy. And immunotherapy, of course, in the oncological setting is really promising, but the placenta, who is key for, or the decidua, who is key for trophoblast invasion, placental development, and plays an important role in the fetal-maternal tolerance, those cells also have these receptors for immunotherapy, and that has consequences. And we have shown that this patient, this child actually received pembrolizumab during pregnancy. The child was born normally, and in green you see the weight of the baby, and you see this sharp decrease. This neonate is dying, and that was due to severe immune-related enteritis. The baby received the treatment as we would give in adults on side effects of immunotherapy, and actually then recovered actually very well. So immunotherapy, that is a challenge of the future on its safety during pregnancy. I'll show some options where we can preserve the pregnancy, but this is not always possible. We see here a high-grade sarcoma, which is really covering the bowel, the peritoneum, also the uterus. We could not save actually neither the mother nor the fetus. This mother basically decided to terminate the pregnancy because she was diagnosed with breast cancer, with bone metastasis and liver metastasis. But important here to tell and to share is that we can decide on termination, but this will not improve the maternal oncological outcome. I promised to tell you something about the follow-up of the children. Of course, we give this chemotherapy while we know that the pregnancy is very protected. Women cannot eat raw meat, raw vegetables, be careful with raw milk, you cannot drink alcohol, you cannot not smoke, but then there is this chemotherapy. Actually, regarding chemotherapy, we have done many studies. This is, for example, in baboons, where we tried to do an umbilical cord sampling after chemotherapy, allowing us to address this question, how about the transplacental transfer? These are our studies in baboon, but other groups have investigated transplacental passage in the mouse, in the rat, and in the perfusion model. And basically, the idea is that most drugs cross the placenta, virtually all in a strongly or moderately or strongly reduced fashion. For example, if you take baboon, you see here 4, 7.5, 1.6, but platin actually in the largest concentration. So there is a passage, but the placenta makes sure it protects to some extent the fetus. Nevertheless, it crosses. So what do we tell our patients? That still we can give chemotherapy. Well, we tell our patients that if you give chemotherapy after 10 weeks, so this is a study on more than 600 cases where we have detailed data on what type of drugs at which moment in the pregnancy. The dotted line is actually the background incidence of congenital malformations. Then from 12 weeks on, we see that actually there are no more, and we know no other malformations when compared to the background, but the risk is increased in the beginning of the pregnancy. So from the second trimester of pregnancy on, we can give chemotherapy. But how do the children afterwards do? Well, I will summarize the data that we have of 20 years research, where we compare children born from mothers who received treatment during pregnancy from children born from mothers with a normal pregnancy. And we have predefined age points that you see here, where we test general health, cognitive function, behavior, and the cardiac function. So it is really a dedicated investigation of the health of these children. And we've shown in several important papers, studies, that actually all these children do well. We've done this in children who are 18 months or older. The New England paper was in children who are 3 years old, but then we continue to follow up the cohort. So 6-year-old, 9-year-old, the most recent data were on 12 and 15 years old, where we also saw for the first time that this pubertal development was also within normal limits. The groups are now large enough to also focus on certain combinations of chemotherapy, like in this paper, platinum and taxanes. And also, for the first time, we could show that even the subset of cohort of children exposed to radiotherapy, that actually all these functions, attention, behavior, IQ, and memory are actually within normal limits, and that those children cannot be discerned from the background population. But chemotherapy crosses, and it has an impact. Here we see that fetal growth restriction, these are the light blue dots, is associated with chemotherapy and the duration. So that's why we need careful follow-up of the perinatologists to follow the growth and to make sure that the chemotherapy is still not too much affecting the fetus. One important exception is cisplatin. We see here cases of overt hearing loss, and all the cases of the hearing loss are actually in the group with cisplatin. Carboplatin, there was only one child with moderate hearing loss. So we try to avoid cisplatin use during pregnancy. This slide shows actually children who do well. They have all been exposed to chemotherapy, and I promise you that they function normally and that parents are very happy with the health of their children. However, there's a high need to continue this kind of studies, especially taking into consideration the tsunami of the new cancer drugs that enter the clinic, and I already showed you the example of immunotherapy. Very briefly on breastfeeding, also in breastfeeding, there is a high need to do more pharmacological studies. We have tried to do that. It is quite difficult. This is an example of 2 patients where we tested doxorubicin, and you see this sharp decrease actually. We have several slides, but I cannot show all of them. But this slide summarizes actually the main results. As you see, a small number of patients, but with doxorubicin, cyclophosphamide, paclitaxel, carbo, and cisplatin. And these are the days until the cumulative relative infant dose is less than 1%, where we think it is safe. And then you see that, for example, for these commonly drugs, 3 days after 2 days, we can actually consider to give chemotherapy and then not to use the milk those days and continue after 2 or 3 days, for example. So it indicates that breastfeeding might be possible in between cycles if the milk is discarded during the first days. But of course, I showed you some case reports. Also here, we'll need much more research to really make this data more solid. Lastly, we have the advisory board. And I want to mention that because it is quite uncommon, and cancer during pregnancy is not an emergency. There is time for deliberation and second opinion, and many colleagues do not have this experience that is needed. So we introduced the advisory board on cancer, infertility, and pregnancy, which is in fact a virtual on-demand multidisciplinary tumor board. You can, you can easily go online without making an account. You can just ask for an advice, as you see here. You complete the basic information, and we try to provide you with an answer within 1 week. We have an overarching international group, and we have several national groups that can work independently, and we offer them the online tool that they can use. So you can really visit the AppSip, and you can also become member. If you want, you can start your own national board. Feel free to contact us. So I hope that I could show you that most cancer treatment options are possible during pregnancy, but it's complex. There are many factors that contribute. That's why this multidisciplinary setting in the referral centers is highly needed. And we really need more pharmacological studies for the new drugs, but also to better test whether breastfeeding and in which situation breastfeeding is actually safe for the child. I need to thank my researchers and all the sponsors, all the patients that contribute to our work. And I want to invite you for the next INSIP conference, which will take place April 2021 in Thessaloniki in Greece. You see the emails. If you're interested, please join us. Having said this, I want to thank you for your attention, and I'm happy to tackle any question. Thank you.
Thank you, Frederick, and thank you, Agnes, for an excellent introduction to the topic. And it's clear that this is not a minor problem that we're facing. If you take the number that have just been provided, there are some 1,800 patients that happen to have both a pregnancy as well as cancer at the same time every year only in the EU already. So that is a major problem from an EU perspective, and likely from a worldwide perspective, even larger. There are questions. If you have any questions, by the way, please insert them to the Q&A button that is down on your screen, not to the chat function. I can see a question that has been entered into the chat, but please enter the questions that you might have into the Q&A session, and we'll pick them up there. For the time being, that one in the chat function will be taken up. Frederick, it looks like, if I read you well, cytotoxics are relatively safe to be given in pregnant patients with cancer, as long as you avoid the first trimester. The data seem to suggest that. And there is no reason to suspect any growth retardation during pregnancy. Is there any data of those kids that were born for the growth after the birth? So is there any growth retardation after birth if the children have been exposed to cytotoxics during pregnancy?
Excellent question. So first of all, indeed, we should avoid the first trimester, but nevertheless, if you look carefully into the slide, the figure that I showed, exposure to the first trimester does not mean that there will be congenital malformation. It increases significantly, but let's say 50% of the children are born normally. So it is not, I mean, because sometimes it happens that a patient did not know that she was pregnant and then she received chemotherapy. Then we have to decide now, what is the risk? So even then we have to say that there is a chance that the child will be born normally. So, but we have to avoid it, huh? But to put it a bit into perspective, and indeed, the more chemotherapy, then your second point, the more chemotherapy we give, so basically the earlier the diagnosis, the earlier we start, the more chemotherapy we will give, and the higher the chance that there will be a reduction in the growth. Well, we see that they have a catch-up growth. It is actually quite difficult to really well, we don't have the data that we can really have, like, for example, every month regularly that we see. But what we see is that at 6 months or 18 months, that they have a normal weight and length. So there is a catch-up growth, definitely. But it's quite difficult to really document this because you need these serial measurements in all children every time. We try to do that, and we have some data, but it was too difficult to really have solid data on that. But if we follow up the children, we see that they perform well, and that then we do these measurements, and then we see that actually the biometry is in fact normal.
Now, the data we have are on cytotoxics, which is logical because they exist already for decades. And the more modern drugs like receptor inhibitors only were introduced some 2 decades ago. These are given in a different way. And you've shown us that, for instance, HER2/neu inhibition is not a good thing to have during pregnancy because of the effect on the kid, the baby. And also tamoxifen has its detrimental effects on the infant. Is there any data on receptor inhibitors during pregnancy and the use of it? Because they will be given daily. They're not given every cyclic like chemotherapy is given. So there is continuous exposure, if there is any exposure during the pregnancy, that may lead to different assessments and different problems at the end of the pregnancy and effect on the children. So is there any data on more chronically given drugs or more daily given drugs, unlike the cytotoxics?
And you mean then targeted drugs like receptor or general?
Well, HER2/neu, HER2 is a receptor. So HER2/neu inhibitor is a inhibitor.
Actually, it is interesting that Herceptin, if you expose, actually, the children are born normally. So it is really the effect really depends on the receptor. And I think that is for all the new drugs, the effect will depend on the passage, which is then determined by different factors. And that's one thing. And the second thing is what is the target, and is this target present in the fetus, yes or no? And I think those two determine actually the effect of new drugs. And we should not forget the placenta, because the placenta also has these receptors. For example, immunotherapy, I didn't have time to show it, but we see that not in all, but in some cases, we see smaller children, smaller placentas in patients exposed to immunotherapy. And that is just because these decidual cells, they contain these receptors for immunotherapy. Other drugs, so the effect will, I think the difference with these new drugs and chemotherapy is that chemotherapy is like old rapidly dividing cells. So that's quite general. For the new drugs where you're referring to, we will have to individualize every, I expect that the effect of every drug will be different, or every group of drugs will be different, depending on its properties and depending then on the fetal targets that are present or not present. So I think the future will be more difficult to really, or we will more individualize to drugs, while we now in the papers, we take all the chemotherapies together. I've shown you the slide with all the studies. Well, we lump all these chemotherapies together. Safety there, so the safety data are there mainly for breast cancer because it's most commonly used. But nevertheless, we group all the cytotoxic drugs together. We will not be able to do that in the future. For example, rituximab, that's something also we see that is used in hematological cancers. Adding to your question, so we see also B-cell depletion at birth. So it crosses the placenta, it has an effect, but also there we know that the baby recovers. We know that the children afterwards, that the B-cell compartment is fine and is normal. So, but there is a temporary decrease as well. So that's why and apart from that, the children are normal. So, but there we see also an effect, clear effect without too many consequences, and that recovers during the further development of the child.
Frederic, the data you have now, I guess, were more or less observational data that from exposing women and getting the results and following cohorts of women. Was there any prospective trial to try to define the efficacy and safety? And shouldn't we envisage now to have some more prospective trial of these anti-cancer drugs?
We follow up all the children, so we prospectively follow them up, and then once we look into the needs and to the number of data and number of patients, and then we try to answer your question. For example, in breast cancer, actually, the numbers are quite solid, but apart from that, well, we need to continue and to use the registry. What we are doing today is in cervical cancer, so there we follow up of the prognosis, because this is lacking information. So we need indeed to continue and to collect more data per organ cancer, per cancer. And
but I'm not talking about following the children here. I'm talking about investigating the drug used in pregnant women. So having prospective trials at that time rather than waiting to see if the children are doing well, which fortunately seems to be the case mostly, because that's exactly my problem. We are waiting to count the problems and then to say, oh, therefore we should not use this one, or we can use this one. And that's exactly the exposing people and then counting catastrophes, which I find not satisfactory. But I know it's not easy. I'm not saying...
I totally agree. But I think you're right. And first and secondly, it's up to us, because I'm afraid that the pharma industry will not do it because it's too risky, and there are maybe legal issues and so on and so on. And that's of course a pity. So we should convince them to incorporate that, because otherwise it will be very difficult. And it's also not so easy to collect finances to investigate this. And that is something I must say that initially we collected, it was really novel and a lot of knowledge gaps. So we could do, thanks to the sponsors, we could do that. But I promise you that today it becomes much more difficult, because we have shown already a lot, but insufficient to address your question. And but then it becomes competitive with other very good grant applications. And then we see that it's become more and more difficult because the gap is a bit smaller than, we filled a big part of the gap, but not everything. but so we are not able.
I can tell you that 20 years ago, we heard exactly the same from companies saying it's not possible. We will harm children. You use them as guinea pigs. And, and actually what has been shown in children, and I hope it's the same in pregnant women, is that they are safer in trial than receiving drugs that have not been tested. And that is, for me, the evidence that we need to think differently. And unfortunately, as I said in my introduction, only the obligation to study in children has led to having more than 400 products approved for children now, which was not the case before. But I want to go to the question we have received. We have one question about what is the risk of malignancy in infants and toddlers or children which have been exposed. Do you have data on that?
Well, we have some data in a way that we don't see any increase of malignancy. We have done, so of course we have to take into consideration that children are not old enough, but nevertheless some acute leukemia, for example, it occurs in young children and we don't see an increase there. From the literature, I know a child, twin pregnancy, and one child had a thyroid carcinoma as a child, but the other child didn't have it.
Yeah.
And then we have, I did not have time to detail on that, but this year we published in Cancer Discovery on genomic alterations in umbilical cord. And we did do single cell analysis. And then we saw a certain signature in cisplatin exposed children. So we still have to see what will be the effect of that. But in all other drugs, we didn't see any effect of in the hematological stem cells, actually. So that is quite reassuring, but we continue to follow the cohort. And so I told you, or the audience, that how detailed we investigated children, but of course we don't do that once they are adults. And then we continue with questionnaires on their health. Until now, we don't see any effect. And addressing actually before your question, and that we need these pharmacological studies. So I can totally agree on that, because I showed you the case with immunotherapy.
Yep.
That patient, it was actually by coincidence, the medical oncologist in the Netherlands had given immunotherapy without warning anybody. So we didn't know about it. It is only when a child became sick that we saw what happened actually. So I totally agree with you that we should have done that prospectively, and then we could document this, and we could also document the growth of the fetus during, we could document everything, and now it's actually too late.
You can limit damage, yeah, if you know.
Yeah. And the child was already actually in a very, very poor condition, and otherwise we should have followed during study, you should follow the child prospectively and very closely, which was now not done. So I totally agree with you.
Yeah. Yep.
Yeah, Frederick, you've shown us data on cytotoxic again, that the passing of the cytotoxic drugs through the placenta is limited. So the placenta somehow serves as a filter, and that the exposure of the fetus is only 4% or something like that as compared to the mother, with some exceptions because the platelets have a larger exposure there. Is there any data already, because this is data that is being collected continuously, I'm sure, in your group of co-investigators. Is there any data like this on the more modern receptor inhibitors? Do they pass the filter? Do they lead to a significant exposure in the kids?
It is not so well investigated, and I must say that it's quite difficult to investigate this, because what we did was in baboons, but that is now 15 years ago. So we went to Kenya. That's one thing. We also used a placental perfusion model, but that's very, I must say, very technically challenging, because you need the temperature, the pressure, really. Any problem will result in unreliable results, and then you have to do that several times to have. So, and then you have to do that, the whole setup, for one drug only. So we definitely need more data, and I think there is definitely a gap, and we need to find a way to investigate this more efficiently. Addressing your question, there are some data. What is actually important is the receptors in the placenta. So some drugs, they need the FC receptor in the placenta, and the placental transport will be determined by the presence of the Fc receptor in the placenta. For example, immunotherapy, we expect that immunotherapy in the beginning of pregnancy is actually safe because there is no receptor and it will not cross. But during the evolution of the pregnancy, the placenta will be larger and larger. There will be more and more Fc receptors that add to the transplacental passage. But apart from that, I must say, many of the drugs, also there is a clear gap. And what we can only do is, or what is the solution, that if new drugs are used, that we test the umbilical cord and we take at birth some blood, because otherwise in humans it's very difficult to obtain actually this fetal blood sampling. And I think there is a clear need to better preclinical models with cell lines, and that we have like a placental model, also maybe a breast milk model to assess that also. I think there is a big gap in and there's an opportunity to further look into that.
But at least there are some models available, animal models. Are these being explored more extensively? Is there any plan within your group or
No.
colleagues?
No. I know a French group that investigates who uses the placental perfusion model, but as I said, it's quite expensive, it's difficult, and it goes very slow because you need a lot of efforts to have data on one single drug. So to be very honest with you, we don't focus on that anymore. Also because we needed to make choices. We are limited in grants, so we have to focus to choose actually what is actually now the most important, and to see, so that actually limits our possibilities. So we did it, but then at a certain time we had to focus and to narrow actually our research field.
Yeah. Do you know if there is any activity ongoing in pharmaceutical industry in this respect? Because if you think of the number of patients, 1,800 per year, every single year in the EU only. I used to treat patients with gastrointestinal stromal tumors, adults, which is a disease that is very popular because there's now 5 drugs on there. And the number of those patients per year is much lower than the number of females with cancer. So is industry considering any use of models in their development? Because is there any plan to be more proactive there? Because there is a need there.
Yeah, I think that would be great, but I think most people know that we are doing research, but the pharma industry is not really interested. They're not approaching me. And when I approach them, it is not in their portfolio. They really have their own portfolio, which is on certain topics. So I'm afraid that the cancer in pregnancy topic is not really in their interest, because when you say,
that's also where regulators have a big responsibility, because they are not asking for the data when they receive an application and they assess it. Although companies are supposed to do it, when they do it, it's not always the case by far, but when they do it, the data are looked at by regulators and put in a cupboard, and the product information is not updated, and you leave the prescriber with do your own benefit risk assessment, on which basis, with no data. So it's really unacceptable. And even at WHO, we've told them to update the essential medicines list, because they are not even inputting the data that exists. They are not perfect, but some of them exist, and they should be reflected in the product information. But this is really leading to we need to have an obligation, unfortunately. Otherwise, it's not going to happen. As I said, it took us 50 years in pediatrics to get the obligation. And when we had the obligation, suddenly we saw the data and the trials were done. Not fast, but they are done. I think we have another question.
Yeah, on fertility.
Yeah, the fertility one, which is a follow-up.
That's a very good question. I think we are busy investigating this. But we have maybe some preliminary data in a way that if we take the pubertal development, secondary sexual characteristics into consideration as like a sign of fertility, then this is reassuring. The boys and girls have a normal pubertal development, normal menstrual menarche, and so on. So it shows that the reproductive system is actually suggests that it's working normal. But we don't have the data in older women to say that they conceive and have normal children, normal pregnancies. We don't have those data yet, but we are planning to do that in a cohort of 50, 60 patients that are old enough to address this question. But
Yeah.
so we don't have solid data, but as far as we have them, it seems quite reassuring.
Yeah. And there is a general decline in fertility anyway, so it will be quite difficult to assess what is the respective role of the cancer treatment and the sort of current decrease. Yeah. We have a question on the, do you think that in silico trials could help resolve some of the questions? Which is, of course, computer-based trials rather than patient-based.
Well, yeah, I think it will. I'm not an expert in the in silico models, but I think they can help. But I know there are cell lines as well, so you could also with cell lines that have with actually in this case from placental or chorial trophoblasts. They have the same receptors actually as the placenta, or more or less. So that could be used as well. So therefore, there are some models available.
Yeah.
In silico, yeah, I'm not really an expert, but I think it depends a bit on the model and how fine, how detailed your in silico model actually captures the in vivo situation.
I think they can expand the number of participants in a trial and give a result with a narrower margin of confidence, because you have more. I mean, they are artificial patients, but they are duplicates.
And that information you could use to decide yes or no to allow them to enter a clinical trial. If that in silico model predicts that the exposure would not be too high, or I mean, you could use that information for the clinical setting. Yeah.
You can certainly also use pharmacodynamic modelization and trying to get as much data as we can from analyzing in a different way from just doing using patient. But I think I'm always interested in the ethical aspect, which for some people say, we all have every one of us as an ethical role to play and to contribute. We should not even have informed consent. We should all contribute because that's an ethical need. It's a bit extreme, but I think it's an interesting project. We need to have more information. Therefore, we need to also maybe ask more often to pregnant women. Would you agree that we measure, we analyze, breast milk analysis, for example, and concentration of medicine could be done much easier than it's more difficult during pregnancy, of course. And
yes, but I think you're right, because what we see is that our patients are really motivated. They see the problem and they want to contribute to it. And I think we're sometimes afraid we should not ask because they are pregnant, but it's the contrary. They are really motivated. If they know that it can help future women, the solidarity is actually enormous. And the motivation of these mothers is also very, very strong. And that's actually part of the success of our study. Part of the success of our study is the mothers who want to continue the pregnancy. And so we should really ask them much more. We should not, they are stronger than we think, and they are more prone to collaboration and to the solidarity aspect.
So I guess that gives us an answer, your answer at least, to the question that was put on in the title of this webinar. I mean, should we by default exclude patients that are pregnant and happen to have cancer from clinical trials of novel agents? And I hear you saying no. Is that correct?
Yeah, of course. Yeah, definitely. And we should not look at it as a paternalistic view. We should, if you ask pregnant women, I mean, you will be surprised how motivated they are, and they will also say, yes, we are happy to join, because we want, we want that future mothers actually will be better served and better reassured about the outcome of studies.
And they may well be more.
Well, we don't have the time, but just wanted to say that also patient organization should be involved, and the cancer patient organization should be involved. Okay, I'm closing here because I know it's time.
It's time to close this webinar. We have a few closing slides, if I can have these up on the screen. Thank you all for joining today. We have future meetings of CDDF being scheduled now, where you can register for already. There's going to be a webinar in just 11 days' time on ACT EU, as you can see here. And if you're interested, please join us then and register for it. We will have our annual conference next year in Ghent, in Belgium, on cancer clinical research and changing global environment. That's a 3-day program in February. If we go to the next slide, there will also be a workshop in Brussels next year, March 30 and March 31st, on challenges in the landscape of global development strategy, including patient access. So how can you design just a few studies that give you all the data that satisfy all the regulatory agencies worldwide? And there will be a meeting in October 5 and 6, again in Brussels, on health technology assessment in cancer drug development. If we go to the next slide, I'd like to thank everybody for your participation. We are always curious to hear how we can learn from our experience. So if you have time, please fill out the survey form that you can access by scanning the QR code that is now on your screen. There's also a link in the chat that you can use for this. Please send us your comments and suggestions. We can't do everything, but we're trying to do as much as we can to get people to receive more information. By that, I would like to thank Professor Frédéric Amand for his excellent contribution. I'd like to thank Agnes Saint-Ramond for her participation and excellent contribution as well. I'd like to thank all of you for your participation to this webinar. And we are looking forward to meeting you again on the next occasion, which will be the next webinar in December. Thank you so much. Have a wonderful evening.